Okay, let's do this. So this is the first figure from the paper. We will get to that. But let's start over here. I should move this to the end. So we're going to be talking about vesicular stomatitis virus. So a good question is, all right, what's this vesicular stomatitis virus? What's it all about? Well, this is going to be what we call a negative strand virus. So the coronavirus is a positive strand virus. And so that just means that all the genes are in there so that they cannot be directly read. What you would have to do in order to be able to translate N, P, M, G, or L genes, this is the genome in order, is you would have to make a reverse complement. So just like a copy, the way that cells make a copy of this, but you'd have to copy RNA and make another set of RNA in order to make a version that could be used. And the virus comes in primed and ready to do this. So L is the polymerase protein. N is the nucleoprotein. So this is going to wrap around the inside, and essentially its job is to protect the RNA. So there you can see the RNA riding in a groove along the top of it. That's N doing its job, and so the RNA is going to be coiled up on the inside of this sort of bullet shape. P is the phosphoprotein. Now we know that P interacts with the polymerase, which is L. The polymerase is there to copy the virus once it gets in there. So these are the virus's own genes, and yeah, it just needs these 5. Anyway, M is called a matrix, so this does the same job as the coronavirus M protein. It's going to be like a bridge between the glycoproteins, or G proteins, on the outside and the nucleoprotein on the inside, which is holding on to the RNA. So the M is the glue that holds the whole thing together. And this virus is actually going to package a copy of its polymerase protein. Something like SARS would not package the polymerase. They would package the gene that codes for the polymerase and then just make it as soon as it got into a cell. So it's just a different way of doing business, but it works out just as well, and it ends up being just as dangerous to human health. Not necessarily more, not necessarily less. It's just a different way to get the same job done. All right, so this is what the protein looks like. The G protein is normally the one that is going to work like a spike and grab onto cells and then do the membrane fusion process. So it actually, G protein is one of the rare ones that does not work just like the coronavirus spike. But most viral fusion proteins actually do work just like the coronavirus spike, at least the fusion proteins of viruses with an envelope, with like a membrane around the outside. This one does have a membrane around the outside, so this sort of light blue shell going around here is the membrane, and it stole this from the cell that it just came out of, so yeah. And yeah, the phosphoprotein interacts with the L polymerase. It acts like a key that makes L polymerase turn on and start to work, and then L polymerase is going to copy the RNA. Now, it's going to be trapped in here at the end of the RNA, just waiting to get started. And then once it gets dropped off into the cytoplasm of the cell, then it's now got nucleotides, and it now just goes as fast as it can and makes copies. It just starts running off copies of the thing that it brought in. So that's the idea. This is a little bit about the virus. Okay, next thing you need to know is, how do we count viruses? So we count them with a thing called a Plaque assay. And so let's look at this one, nice and big. Here we go. So a plaque is like a hole. That's literally all it is. And this works when you have a virus that will go into cells and will start to kill those cells. Given enough time, the virus will actually clear out a little section. So if you put one virus in, it'll kill that cell. Then it'll spread the cells around it. It'll kill those. While it's spreading to the cells around that, it'll kill those. And it turns out if you leave this for something like 2 to 3 days, you'll get a nice round clear hole in a layer of cells. So the pink stuff down here is a layer of whatever cells the virus likes to grow in, and the little hole in that is something that has been punched by one virus. And so this is the way you count working copies of the virus. You take your virus, which you don't know how much is in here, but you're going to make dilutions of it. So you'll have just like the straight up virus, 1/10 that concentration, 1/100th, 1000th, 10,000th, etc., like this. And you're going to then put these onto what we call monolayers. It's just a layer of cells that the virus likes to bind to and fuse with and grow in. And they're in a dish of some sort. Or sometimes you'll see them all lined up together in a 6-well plate like this guy, or a 12-well plate like this guy, or you can do it in even smaller plates if you're crazy like that. And so then you're going to count how many little holes you make. And so if you have, let's see, 2 times 10 to the 7th virus particles in this tube over here, then by the time you dilute it by 10 to the 7th, you should be left with 2 virus particles. Yeah. And so in the one that you diluted that's like 10 times as concentrated over here, you should have 20. And in the one that's 10 times as concentrated as that, so if 2 is here, you should have 20 there because that's 10 times as concentrated. And how many should you have here? 200, right? And eventually you can keep doing these for all of them, but eventually you'll get up to a point where you just can't read it anymore. It's just too many little holes and they start to overlap with each other. So you usually want to find whichever well has somewhere between like 2 and 20 or 5 and 50, somewhere in that range. And that's the one that you want to read out. And you normally do this in multiple copies at the same time so that you get a good average of how much virus there is. And so, like I was saying in the chat, this is like counting bombs by the craters that they leave. Just, you drop them and then say, oh, yeah, there's 20 holes down there now, so there must have been 20 bombs. Yeah. You don't know how many bombs were duds, because those aren't going to leave a hole. But at the same time, if they're going to be duds, then what does it matter if we count them? That's kind of the idea here. And anyway, this is just how you do a plaque assay. We don't get to choose the way that it works. It just happens to be a method that's pretty good. And so then when you're done, to read it out, you put this nice crystal violet stain on there, which is just one of the prettiest colors in the world. And that'll stain up all the cells. It binds to the glycans on the surface of the cells. And yeah, the little holes that you see there are places where the cells got blown up. And so you just count that number of holes, and that'll tell you exactly how many viruses you started with. Okay, so with that, we can now start to pick apart what is in figure 1 over here. So figure 1A, you see those genes? What do those look like? We've got an L, a G, an M, a P, and an N. And for some reason, it looks like they stuck in enhanced green fluorescent protein, which is a more stable version of green fluorescent protein. The original jellyfish version isn't designed to stay on forever. The jellyfish doesn't necessarily want to have like unlimited fluorescence. But people messed around with it in the lab and they got enhanced green fluorescent protein, and that's really good. Yeah, nice and bright, and it real stable, just stays there. Okay, and so that is actually, of course, that's the genome. Yeah, look at that. So NPMGL, and over here NPMGL, just like that. And at either end, we have a leader or a trailer RNA, which doesn't code for anything, but these are important. These are things that the polymerase is going to recognize, and that's how the polymerase is going to know to copy this piece of RNA, not just any piece of RNA. And so these are the viruses they're working with. So they have taken the virus, they've stuffed GFP into the virus, and they actually did this many, many years ago. And then they've got the GFP version of the virus, so they'll be able to tell where it is because it fluoresces. And they also put in, instead of the G protein, which is like the equivalent of spike, they just literally put in the SARS coronavirus 2 spike. So right in this region, let's get off of there. There we go. All right. And then they also tried to put in this thing called spike delta 21, which is missing 21 amino acids at the very tail end of the spike. And so they'll tell you in the paper exactly why they did that. I want to see, see what you think. But these are things that are useful for the coronavirus. This is the idea. They're going to interact with coronavirus proteins, but they actually impede the interaction with VSV proteins, because this is got to interact with the VSV M protein and N protein potentially if it wants to get incorporated into the virion, and that's totally the goal here. So that's what they did. This is the growth of those viruses. So you can see that the virus gets in, and these are plaque assays. And so in this particular plaque assay, the plaques are dark and the background is light. That's a different way that you can do it. But yeah, it worked out very nicely. And so they tried putting in different proteases, for example, or different genes to help try to activate the virus. And these MA104 cells did it very well. And if you look over here, though, the SARS coronavirus 2 delta 21 seems to give a much better set of plaques. So these are the plaques at the same dilution. And so if you've got 50 to 100 plaques in the bottom row and 50 small plaques in the top row, that means that the top row one got in at the same rate, but it didn't spread at the same rate once it was in to the cells, once that first wave was done. So these are all things, they're checking up. They're checking up to make sure that everything goes right. And these are being done in vero cells, which are those ones from the African green monkey, Cercopithecus aethiops. Okay, and so shown over here in panel C, these are a bunch of different cell lines. A549 is a human lung cell line, but it's a lung cancer. BHK21s are from baby hamster kidneys, and I forget what the 21 is. It's just the isolate number, I think. KECO2s are from colon cancer, if I remember right. Cancer of the colon, I think that's what those are. The HEK293 and 293Ts are human embryo kidney cells. So those are good. HeLa cells are from a human, and these are a cervical cancer cell line. And you got a bunch of other ones. HepG2, I forget. I think that is a hepatocyte line. I don't remember what HT29s are. HU87s are definitely hepatocytes. So these are cells from the liver. MA104s, I know they use for some DNA viruses, and I do not remember where they're from. The Vero with the CCL81, that's the Vero cells. Vero E6 is another Vero cell line, and Veros with furin with an extra protease stuck in there. That's another version of that. And you can see the ones that it gets into best are the MA104s, and second best would be the Vero E6 line. So there we go. And they're showing over here titer. Okay, titer is PFU per mil. That's what that means. And so titer means the amount of virus that we can count in a sample. And so PFU is plaque forming units, because we don't know how many viruses there necessarily were. If 1 out of 10 was working, then there were 10 times as many viruses. If every single virus works, then this is an exact count of how many virions, virus particles, there were. We don't know that, so we just say it's this many plaque-forming units. So think like active viruses that worked in the system, they happen to be trying them on. And that's per mil. And so there we go. So that's this virus getting in and growing quite nicely, particularly nicely in those MA104 cells. And down here at the bottom, they're showing, well, what if we had Vero cells and we took the CCL81 version or the E6 version, which we know are both okay. And then what if we put in Tempris 2, which you'll probably remember is the thing that cleaves right before the fusion peptide in spike. And so they are vastly overexpressing this protein, which is like the activator for the virus, on every single cell. And so what do you think that would do? Well, I think that would lead to more infection and more spread. And and so what it does is you've got definitely more spread. Look at the size of the plaques here. They're like the size of a dime, whereas the plaques over here are the size of, I don't know, the back end of a pin maybe, but quite small. So I'm comparing these guys to these guys. And here we'll back off. There we go. Now you can see it. Okay. And over here in panel E, they're showing that, well, did we actually make the protein we set out to make? And so VSV is just unmodified VSV, which doesn't have a spike protein. Over on the other side is the VSV expressing the spike protein. And as you can see, they were able to make it. So they're going in here. This is a thing called a Western blot, where they use an antibody against spike to show you where the spike is on the gel. That's a nice little technique that you probably have run into several times. And if not, just look up Western blot. It's got a good Wikipedia page. It's not tough. It's it's a pretty easy technique. And then shown over here, so E, they were looking for specific proteins, and F, it looks like they're looking for any old virus protein. And what they've got in F is they've got all the normal proteins of Vesicular Stomatitis Virus, including the star band, which I think might be a either a cleavage product or a super glycosylated version. Let's check. In panel F, okay, monk infected, labeled. Oh, okay, that's easy. And analyzed. Asterisk indicates a band that was also detected in the mock lane. Okay, so the band that's here is something that doesn't mean anything. This was also detected. This is something that the virus does to the cell. So what they've done is they've gone in and they took cells, they added actinomycin D. And so this is going to stop the cell from being able to make any of its own messenger RNA. So the cell is going to be just like on pause, but it doesn't block virus replication from working. And so the cell's polymerases are going to be off, but the virus's polymerases are going to be super on because the virus brought its own polymerase, the L protein up here. And so this is looking at, well, what's the L making? What size are they? And sure enough, it's making L protein. There we go. And P and N and M, just right. But when they put in the G protein in normal VSV, yep, it makes the G protein. And when they put in the spike protein over here, then yes, you get the spike protein. I have no idea why they get that extra band there and they don't get it over here. That's a little weird, but this is apparently something they have seen before. Okay, and then finally, in panel G, they're showing you, actually, we better look at this zoomed in again. There we go. So this is what the vesicular stomatitis G protein looks like. This is what the spike protein looks like. And the bottom bits where it gets a little weird, those are the parts that they haven't solved the structure for. That's when we get close to the transmembrane. That's so far been a part that they have to model and they can't actually solve. But you can see that the viruses do look like little bullets, and the regular VSV has a row of little tiny bumps on the outside. There are these little short VSV G proteins. And the SARS coronavirus 2 version of VSV, look at that. It's covered in little, well, actually quite big coronavirus spikes. And so they did it. They made a virus that makes its own SARS coronavirus spikes in place of the normal VSV G protein. And that's what they're using in the rest of this study. And this study doesn't go into a lot of immunological detail. Mostly they're looking at how this thing works on a basic level. But yeah, it's useful. And yeah, so have a look at the rest of the paper. This is a nice little introduction to live virus vectors. Basically using the parts of a virus to do the jobs they normally do, but as part of a vaccine rather than just part of the virus growing itself in the world. So there we go.